Welcome back for our final segment on our lecture series on glycolysis. So the goal here is to really dig into the idea of these regulatory enzymes. Like, how do we control glycolysis, or how does our body control glycolysis to run the cycle more when we need it, less when we don't? And as part of that, we are going to compare these regulatory enzymes in the liver and muscle cells. So just to recall, glycolysis is one small part of the tons of pathways in metabolism. And this small part, remember, we are starting with glucose, ending with pyruvate. And a key idea here is that the enzymes that catalyze irreversible reactions in the metabolic pathways are potential control sites. So those irreversible reactions, the enzymes that control those, can control the speed of our reactions. Now, this is small in here, but I can look at it and say, wow, okay, here, this is a two-way arrow. But there are plenty of single direction arrows. Here's one. And so those single direction irreversible ones are the ones we're looking at. So if we go back to this summary slide and you look at these arrows, we see we've got step 1, is an irreversible reaction. And if you remember, we're phosphorylating that glucose and then it's kind of trapped in the cell. All right, step 3 with our phosphofructokinase, that PFK enzyme. And step 10 are the ones that tend to be regulatory because they're reactions that go only one direction. Okay, the three enzymes that are involved in those. So here's step 1, step 3, step 10. Those are the three we're really going to be looking at. Now, one thing I want to point out is that control mechanisms are different in different cells. We have looked at this term before, I believe, isozyme. If not, learn it now. So it's different forms of an enzyme. So different forms of an enzyme, but they catalyze the same reaction. So essentially, the regulation of these 3 steps, step 1, step 3, step 10, the regulation can differ cell to cell. Okay, and it has to, based on the other types of chemistry that are happening in the cell, what metabolites are available. Okay, and so we'll look at that. We'll look at how, for example, the amount of ATP in the cell has a much bigger effect on muscle cells as opposed to liver, in part because of this other types of chemistry. Anyway, we're going to take a look at all this, and we are going to compare glycolysis regulation in muscle versus the liver. So let's look at regulation of glycolysis in muscle. And so just to kind of remind you, we're looking at these 3 main regulatory enzymes: hexokinase, step 1, PFK, step 3, pyruvate kinase in step 10. But it turns out that this PFK provides a major regulatory control. So the hexokinase in step 1 is important, but there is another pathway for that glucose 6-phosphate. Step 1 makes glucose 6-phosphate, and it can either continue on or it can get ramped off into glycogen. So the PFK step is the first step, that step 3, after which everything essentially continues on down to the product. So all the reaction that PFK pushes through should continue down to finish out our glycolysis. Okay, if we think about muscle, the major role of glycolysis in muscle is to provide ATP for muscle contraction. So when we are at rest, Okay, we have relaxed muscle fibers and we're not trying to contract them. So when we're at rest, we really want those 3 regulatory enzymes to be muted. We really don't want them speeding up that reaction because we don't need that energy. It would be wasted on us. Okay, so they have these negative effectors. But when we're exercising and we need all that ATP, we want these to be expressed. We want these enzymes to really ramp up their work and speed up these reactions. And so those enzymes will have positive effectors. And we're going to look at these two separately. So let's start with when we're at rest. So when we look, you can see right here, for example, glucose 6-phosphate is a negative effector for hexokinase. So you see that negative sign. And that makes sense if you think about it. If glucose 6-phosphate's building up, it means it isn't getting used, so the rest of this reaction must not be proceeding that fast. And we need to slow down this step. So glucose 6-phosphate is a negative feedback effector for that hexokinase. Then, if we come down here, we said this PFK was a key regulatory one. We are going to look at the ratio of ATP to AMP. So we're used to seeing ADP, that adenosine di, but this one's the mono one, and we'll look at why, okay? But essentially, this is called the energy charge. So energy charge is that ratio, okay? And if that number is high, it means, hey, there is a lot of ATP already. We need to slow down these reactions. And so when that happens, that is a negative effector for both the PFK and the pyruvate kinase. It slows down both step 3 and step 10. And I just wanted to point out over here that in step 3, the one that is catalyzed by PFK, we use ATP. So we have the fructose 6-phosphate reacting with ATP to give us this bisphosphate molecule. When that happens, this ATP bonds in the active site. So we have both the fructose 6-phosphate and the ATP interacting with the active binding site. But now we're also saying that ATP, besides being used in the reaction, ATP is also a negative effector. And so there would be a separate binding site where those allosteric effectors bond, so where the ATP would bond, that would slow this enzyme down. Okay, let's take a look at what happens during exercise. Let's start with the purple one. So again, we're going to look at that energy charge, the ratio of ATP, right? So energy charge, ratio of ATP to AMP. But now a low energy charge is what's going to be our signal. Low energy charge means, wow, there is not much ATP, and it's going to come in and it is going to speed up this reaction. That is a positive effector for the PFK. Then, if you think about this, as the PFK speeds up reaction 3, we are going to see more of this fructose 1,6-bisphosphate made. So we start ramping up the amount of this. Now there's a bunch of steps here, but the amount of this will help drive those steps forward, okay? And in part because that last regulatory enzyme, pyruvate kinase, has an allosteric binding site for this molecule. So lots of this molecule will just continue through our reactions. But some of it, as it's floating around, will bind to this enzyme, and that starts speeding up this process. And as that happens, it starts pulling, think Le Chatelier's principle, it starts pulling all of these things forward. So glycolysis in muscle, right? We're talking about muscle right now, is regulated by feedback, so we saw that, and by the energy charge to meet the need for ATP in the cell. We said that PFK is a key regulator, but the entire pathway is regulated by feedback at different enzymes. And I said we'd come back to this. PFK is allosterically inhibited by ATP and allosterically stimulated by AMP. And we said, well, why this? Why not the ADP? That's the one we're used to talking about. But when we really need ATP, we get this reaction occurring where two ADPs react. One gives a phosphate to the other, so we end up with ATP that we can use for energy, and we end up with that AMP. So when we see this AMP start ramping up right here, that tells us that, wow, we must be really needing energy because we are reacting our ADP to form ATP, and that tells us that, hey, we need to speed up this reaction. If we look at our PFK enzyme, so it is a homotetramer. It's got 4 strands. You see that in this ribbon picture with the green, yellow, blue, and red. Okay, they have allosteric sites where things can bind to speed up or slow down the process. And the catalytic sites where our molecules, the ATP and our other metabolite, will bond to move forward with the reaction. When we have high ATP, we said that when extra ATP bonds in these allosteric sites, when we have high ATP, it's an inhibitor. And we see this curve shift to the right, and it's very sigmoidal. But when we have really low ATP, we see that it's more, what's the word I want? But we see the curve that is less allosteric, right? We start shifting back to the left. Okay, so that was in the muscle. Let's talk about regulation of glycolysis in the liver. This is actually much more complicated, largely because the liver has a lot more jobs. Muscles just need to move for the most part, but the liver has a lot of jobs. And so the regulation needs to correspond to this biochemical versatility of the liver. The liver does a lot. The same 3 enzymes are going to be important for the same reasons, but they're going to be controlled a little differently. As we talk about regulation of PFK, so we're going to start with that main one, in the liver, the key regulators are going to be citrate and fructose 2,6-bisphosphate. Citrate is a metabolite from early in the citric acid cycle. So remember that our pyruvate, which is the end of our glycolysis, is going to eventually react into molecules that go into our citric acid cycle. And citrate is a piece of that. And if that citrate can be used, so the metabolites in that citric acid cycle help carry the electrons down for use in the electron transport chain. They're used as starting materials for biosynthesis. So if citrate is building up, it's meaning it's not needed for those other things, right? It means that our biosynthetic needs of the cell are already being met. And so this is a negative effector. It slows the process down. On the other hand, we have this molecule fructose 2,6-bisphosphate that is an activator. And I just want to point, and we're going to look at this on the next slide, that this is not the same molecule that is in our glycolysis mechanism. That's fructose 1,6-bisphosphate. Before we do that, real quick, energy charge, just like in the muscle, is also an effector, but it's not very important here because in the muscle, that ratio of ATP to AMP is changing dramatically depending on what we're doing, right? Are we resting? Are we jogging? Are we running from some predator? So that ratio is changing, but the energy needs of the liver are more stable. And so the energy charge doesn't change very much. Okay, let's come back to this picture. Here's that third step of our mechanism. We have fructose 6-phosphate reacting with ATP with our phosphofructokinase, PFK, to give us fructo-1,6-bisphosphate plus the ATP and proton. So here is the metabolite in our glycolysis. And then the positive effector is this fructose 2,6-bisphosphate. So notice here that here this phosphate is on the first carbon. And it might look a little confusing here, but just because we've drawn it in a different direction, that first carbon's down here. And notice it doesn't have that, but instead it's bound through that second carbon. When there is a lot of this molecule, it indicates that there's high blood glucose levels. So anyway, let's take a look at why. So in that reaction, we had the, right here, fructose 6-phosphate as our reactant. And in that reaction, moderated by that PFK molecule, we know we make that fructose 1,6-bisphosphate. But if some of this builds up, okay, so if those early reactions are going really fast and we start seeing this build up, some of it gets directed to another enzyme, okay, called PFK, whoops, can't see that very well, PFK2. And that enzyme converts it to a similar but different molecule, this molecule right here. And that molecule is an effector of PFK, and it's a positive effector. Okay? Essentially, if our reaction is just going along great and reaction 3, this is reaction number 3 here, is moving along at an appropriate speed, then there won't be a lot of this built up, and so we won't make much of this fructose 2,6-bisphosphate. But if the early reactions are moving faster and then this is starting to build up because our reaction 3 is not moving fast, that's when we're going to start making this. And so then this serves as an allosteric effector for the PFK to tell the PFK, like, get moving, come on, get this going, let's speed up this reaction. So when we look at this, we only are showing this reaction in one direction, but really this can go in both directions. And this is just an interesting enzyme because the enzyme for that reaction can catalyze in both directions, which means it's called a bifunctional enzyme. And it has different names depending on the direction it's catalyzing. So when you look at this, it has one single polypeptide chain. Okay, this enzyme is one single polypeptide chain, so no quaternary structure. It's got about 470 amino acids. Half of it is a kinase domain, so the part you see in the purple over here, and the other is a phosphatase domain. So the kinase will add a phosphate, the phosphatase will remove a phosphate. Okay, and if it's working as the kinase, we call it PFK2. But when it's working in reverse as a phosphatase, we call it fructose bisphosphatase, so FBPase2. Okay, so you don't need to memorize those names, but you do need to kind of understand the idea that, hey, we've got this, essentially this second route that fructose 6-phosphate can react, and that tells our enzyme, wow, hey, we're not moving fast enough. We're building up this side product. right, let's talk about that last one, step 10, the oh, I'm sorry, not step 10, the first step, hexokinase for step 1. So when we look at that hexokinase, it's an allosteric enzyme in the liver, just like it was in the muscle. And remember, this is the one primarily responsible for phosphorylating glucose. And the one in glucose, we have a special name for it, right, is glucokinase or hexokinase 4. So we talked about the idea of an isozyme. So this is just the version of that enzyme that works in the liver. And it's kind of important here because it's only active after a meal when the blood glucose levels are high. And that's important because we don't want the liver to pull glucose from the bloodstream when other organs need sugar. So we're only going to activate this essentially when blood glucose is high. All right, let's talk about that. Reaction 10, the pyruvate kinase regulated reaction. So the pyruvate kinase reaction in the liver is regulated allosterically, as it is in the muscle, but it's also regulated by covalent modification. So we're going to look at that. So when we look at this, pyruvate kinase has allosteric regulators. So those are the ones in this oval down here. So that fructose 1,6-bisphosphate is still the allosteric regulator. Okay, speeds it up. We looked at that before. ATP, allosteric regulator that would slow it down. Okay, if we've got a ton of ATP, then that tells our system, hey, wait, we don't need to keep making more. And interestingly, alanine. And I put a little note over here because, why in the world alanine? That alanine is very close to pyruvate, and so pyruvate can be converted to alanine. So if we start building up this pyruvate, and we don't have anything to do with it, some of it can get converted to alanine, and hence alanine is a negative effector because if we start building it up, it means, hey, we've got too much of our product. We should slow it down. All right, when we look up here, so we have two versions of our pyruvate kinase, the phosphorylated one, shown in pink, and the dephosphorylated one, shown in green. When we have really high blood glucose levels, okay, that activates our hormone insulin, and it dephosphorylates, right? We're going to remove the phosphate group from this to have the more active version. On the other hand, we do that for a while, and then say our blood glucose gets pretty low, glucagon. We have a different hormone that's going to help us use ATP to phosphorylate this and inactivate it so that we quit using up that glucose. All right, here's a question for you. So which of the following is not an intermediate in the glycolytic pathway by which glucose is oxidized to pyruvate? And there is a reason that this question is here and it was not in our first video or our second video or our third video. But the reason is we have looked at these reactions over and over and over. Okay, and so by now you should be familiar with the metabolites, like what are the reactants in each of those steps, the products. So you should be familiar with those. You should be familiar with the enzymes. You should be familiar with the regulation. So when I look at this, I'm going to start thinking about, which one is it? And when I go back through, having thought about it, I'll find that actually this is my answer because it's actually glucose 6-phosphate is our metabolite. So by this point now, you should really be able to go back. Let's go back to way back to our piece. You'll remember this from the last video too. you should be able to come back here and really step through these. I like the table in the first video best, but you should be able to step through them. I'm not going to probably ask you to draw these, but I would expect you to be able to identify, like, what are the reactants of the first step? What are the products? What happens next, right? You're going to know that, oh, we're phosphorylating glucose in the first step, and then what happens in the next step, that type of thing. All right, that is it. We have finished all the glycolysis videos. Congratulations.